liver seminars from Santander were Joaquín Cabezas and Javier Crespo. Many thanks to Rafa Bañares and Jordi Gracia. Thanks for your allowing us to host the Cyber seminar from Santander. It's a pleasure and an honor for us, and especially for having a person like Ramón Bataller. Thanks, Ramón. Ramón Bataller graduated from medicine at the University of Valencia in 1989, completed the residency in gastroenterology in the Hospital Clinic of Barcelona in 1994, and performed his doctoral thesis on the role of the vasoactive substance in liver fibrosis. Within 2000 to 2003, we completed a research fellowship in hepatology at University North Carolina at Chapel Hill with Professor David Brenner. During this period, he investigated the role of angiotensin II in liver fibrosis. From 2003 to 2011, he was senior physician at the liver unit of the Hospital Clinic in Barcelona and coordinated the management of patients with alcoholic liver disease. During this period, he performed several translational studies in alcoholic hepatitis and identified several novel targets for therapy. In 2011, he joined the University of North Carolina and led an international effort to develop the first histological classification of alcoholic hepatitis and describe the role of systemic inflammation of renal dysfunction. Later, it was the overall PA on an international consortium to study alcohol hepatitis in team. Recently, he is serving as chief of hepatology at the University of Pittsburgh Medical Center. He is co-pilot of a new work that involves translational studies and clinical trials. Website, clinical and research activities amongst very active in the national and international arena with multiple international talk on alcoholic hepatitis, and he serves as chair of the special interest group for alcoholic liver disease in the American Association for the Study of Liver Disease. And during the last years, he was working in integrated omics in patients with alcoholic liver disease. Moreover, he recently created an international network to study alcoholic hepatitis and has generated several studies using big data. And finally, in addition to all this impressive resume curriculum vitae, Ramón is an excellent physician, a friend of his friends, and a fantastic speaker. And then Ramón will give the conference integrated multiomic for the identification of novel therapies for alcoholic hepatitis. Well, Ramón, thank you very much.
Thank you so much, Javier, for this kind introduction of words. I try to be to be at the level of your words. Thank you, Edibabs, and all the organizers. It's for me a pleasure also to talk in this session because, as you may know, probably in a few months I'm leading to Spain again, so I will work more closely with you. Well, in the next 45 minutes, I want to talk about part of my research that has been to do basically omics in human samples to identify targets for therapy, molecules, pathways that correlate with survival, and identify potential targets for therapy. And I would like to combine my clinical expertise with my PhD part, my research expertise, to jump from clinical questions to research approaches, etc. So you understand a little bit why we do things, what is the potential applicability of our findings. So let's start with the natural history of alcoholic liver disease. You all know it. It's very similar to NAFLD. When you drink a lot, you develop a steatosis, fat in the liver, ASH or steatohepatitis, fibrosis. You can develop a cirrhosis, liver cancer, decompensations. But different from NAFLD, patients with advanced fibrosis, many of them cirrhotic, many of them with advanced cirrhotics, but some of them with immature cirrhosis or even non-cirrhosis. When they drink very much, they can develop a syndrome or a disease called alcoholic hepatitis or alcohol-induced hepatitis. That is a rapid development of jaundice. The patients die very frequently. say steatosis, fibrosis, steatopatitis, cirrhosis, but some of the patients that drink very heavily with advanced fibrosis or cirrhosis that can develop this syndrome of this disease called alcoholic hepatitis, which is unique. And most of the attention in alcoholic liver disease has been paid in this particular phenotype, okay? So this phenotype is very frequent in America and the UK, a little less in continental Europe. We have 80% of our patients hospitalized right now in the USA, in the Pittsburgh area, have ALHEP. It's a massive epidemics of this, has been increased with COVID. It affects to young ladies in many of the countries. And I will tell you what is the status of the therapy. This is, I don't want to depress you guys, but this is a summarizing slide of the only approved therapy, prednisolone, for this disease. It was proposed when I was 4 in 71, and it was confirmed in 15 that increases 1 month survival. Beyond 1 month doesn't have any effect on survival. So the only therapy that we have for this disease was proposed more than 50 years ago and only increases 1 month survival, which is okay, but it's not enough, as you can imagine. So we need to develop new targeted therapies for this condition. This is a slide that I will stay a little bit because this is my vision of translational research in this disease. This can be applied for many diseases. And to start with, say, ideally, translational research should start in humans and should finish in humans, ideally. Start in humans means that you have well-described and well-designed databases with all clinical parameters. You get biospecimens as many compartments as possible. Ideally, if from one patient you get a lot of compartments and you can do a lot of omics at different layers, is what we call the superomics. We're doing superomics, patients who are able to, we can obtain a lot of compartments from tissues and we can do a lot of layers of omics. These correlations of these omics in a single patient are much more powerful when you do only one layer in one compartment and you compare different patients, but you don't have all the data from the same patient. So first message today in my talk, the superomics, these patients that I can get as much information as possible, that is the way to go, in my opinion, though they are not easy, okay? So once I do integrated omics studies and I have these 6 subtype, I know the prognosis, I do correlations. And we need systems biology people capable to integrate different omics, to correlate with biological data. So we identify which pathways, which molecules, which biomarkers are associated or not with survival. But when something is associated with an outcome, I don't know if it's a player or it's just a biomarker. How to differentiate a target for a biomarker to do functional studies. And here I had to say that at least in my field in alcoholic hepatitis, the mice models have been very disappointed. Rodents don't like alcohol, metabolize alcohol very quickly. They don't develop liver failure due to alcohol. They can develop, for chicken wire fibrosis, if you do alcohol with DDCs and with CCL4. We have worked many years to develop such model. We don't have a good model. That's why human-based tools like Liver on a Chip, organoids, I know Pau and Jordi, many people in EDBAPS are working on that. It's probably the way to go when mice do not reproduce this disease exactly. Be careful when you jump from suboptimal mice models to human studies, clinical trials. I will show you one example in the next 2 slides. That is very dangerous. Shortcuts in life and in science are good. And I will always say, if you identify something in an animal model, before you jump to phase 1 of 4 clinical trials, confirm that in translational studies in human samples before you close the deal. Obviously, if a functional study reveals that a molecule plays a role in the progression of the severity of a model of the disease, we can close the hook with a preclinical study, clinical trials, etc. So this is the most rational way to do it. Let's have one example of a shortcut. In alcoholic hepatitis, when I was studying medicine or when I was in my residency, they always told me alcoholic hepatitis is due to a PMNs in the liver, is a exacerbation of hepatic inflammation. Therefore, prednisolone or other blockers of inflammation like TNF blockers, for example, would be fantastic in this disease. They will cure the disease. So that was due, the TNF alpha as a target was only relayed in animal models. And all these animal models that led to all these hypotheses, I'm not going into detail, were based in models of mild liver disease, or models of early ASH, or models that didn't imply at all liver failure or portal hypertension. So what happens when we block TNF alpha and ALHEP based on the studies of subclinical or preclinical models where we're suboptimal, did not reproduce the main findings of our disease. What happens that when you have a shortcut in life and in science, the results are not good. And when this is the trial that from the Mayo Clinic that basically finished this story of TNF blockers, basically when you block TNF, you increase mortality more than double because basically infections. When we critically review which are the biological properties of TNF that explain the results, we have two explanations. TNF is essential for liver regeneration. The NF-kappa B, probably the pathway more activated by TNF, is a lethal embryonic knockout because it's a massive apoptosis of the liver. Cytokines are essentially including 6, for example, for liver regeneration. Be careful when you block a cytokine in a critically ill patient, also for the second reason, bacterial infections. We know and we learn from the IBD world that blocking TNF favors a lot of infections. And the main cause of mortality or progression of the disease and development of multi-organ failure is infection. So we were blocking something without checking that in humans that some of the biological properties were dangerous. Okay. When we started to do translation in the years 2000s, and this is from Jordi Colmenero and Marlene Dominguez in our lab, we showed that TNF was not even upregulated in the liver. We found that rather CXC cytokines like GRO-alpha and interleukin-8, and now there is a Nature paper from the BASA group confirmed that, they correlate much better with survival. So we were blocking TNF, something potentially dangerous that was not even upregulated or dysregulated in the liver of these patients. Okay. So another thing that we did, this is a multinational effort to develop a specific histological classification for ALHEP. We found something unexpected, although Dr. Bruguera, a retired pathologist, found it many decades ago. We found the following: the more PMNs you have in the liver, the better you do. Before that study, when I saw this patient, a lot of PMNs around hepatocytes and ductal reaction cells, etc., I felt this guy is going to die. And it was the opposite. This person has 4 times more chances to survive compared to a patient with less PMN infiltration. So this paradigm for the 70s, that is all PMNs driven, is all exacerbation of inflammation of ALD, it was not clear to us. Maybe the other important determinants, maybe regeneration of function of the hepatocytes are really impaired. Maybe systemic inflammation is driving multi-organ failure. That's why that led us to a subset of studies. I want to show you here one of the recent studies that we did. As a doctor, and I'm sure the MDs that see me now will agree, you see patients that drink like crazy, that develop ASH, a little steatohepatitis with some cirrhosis, fibrosis, even cirrhosis, but the liver never fails, they never get yellow. Versus others that drinking the same or even less, they develop ALHEP. Why some people with never decompensated ASH are so resistant compared to the ALHEP that fail? We did a multinational study, it was published recently, comparing a huge cohort of patients with never decompensated ALD, basically ASH with some fibrosis, versus ALHEP. This study was led by Meritxell Ventura, and that is now in the BioBron. So these are, let's see these quantitative things. So never decompensated have similar steatosis. Alcoholic hepatitis have more fibrosis. The chicken wire is crazy. Remember that the fibrosis classification is very hepatitis C-centric. The METABIR was not meant for alcoholic hepatitis or NASH. We see patients with chicken wire, this kind of, you see here, this chicken wire, with a full developed cirrhosis with a portal hypertension of 20. Can you imagine a hepatitis C with an F2 with a 20 of portal pressure? Impossible, no? So that's why the SAVA, for example, have proposed a new classification of fibrosis. The METABIR, remember, very virus-centric. We need to to transform these classifications to more fatty-centric, okay, more friendly for the fatty liver disease. So PMN infiltration was more than ALHEP, but this to me was the most interesting finding, where I put the arrow. This is hepatocellular cholestasis, canalicular and ductal bilirubinostasis. Obviously, patients with jaundice, they are yellow. The canalicles, the ductals do not dry the bile efficiently. But we found there's a subset, 10% of 12% of patients with early ASH that they were never yellow. When you do the biopsy, the hepatocytes were starting to accumulate bilirubin or bilirubinostasis. Is that a predictor that these patients are close to decompensate? We're doing in fixed tissue right now, and with Maria Hernandez and our friends from Caroline Lagne, etc., we're starting a study using all samples of patients that decompensated later on to see whether we can find like a dysplasia, something molecular in early alcoholic liver disease that can tell us you are still compensated, but there is signs in your hepatocytes. There are methylation. I will show you later the rationale for that. Your hepatocytes are starting to dysfunction molecularly. You are likely to decompensate in the 2 years if you don't have abstinence, if you don't reach abstinence. It's like the concept of the dysplasia for a polyp. So I think biopsies and histology in hepatology need to provide more molecular predictors, more molecular signs. Otherwise, just for hematocylin, eosin, and for trichromate, we are leaving a lot of alcoholics with a liver biopsy. We need more prognostic, more molecular information for personalized medicine. When we did the histological analysis of progenitor cells of ductal reaction cells, and I want to see, I think one figure was more than 1,000 words, no? And you can see that this is a silent or never decompensated ASH, and this is an ALHEP. Look at this proliferation of these cells. It's not crazy. When you see these patients in the explants, I will show you a picture in few slides. The patients that undergo early liver transplant because they were not responded to corticosteroids, it looks at pseudocholangiocarcinoma. It's crazy. You think this is only a surrogate bad outcome? I will show you that it's a surrogate bad outcome. This is an inefficient attempt to regenerate the liver, but such a massive amount of ductules that they are not driving the bile. You don't see bile inside the tubules. They are probably obstructing blood flow. They are not only a marker of bad outcome, they are contributing to the patient's poor outcome. And we have some working hypotheses that I want to share with you. So at the molecular level, when you compare here normal liver, silent ASH with alcoholic hepatitis, my goodness, alcoholic hepatitis is a completely different world. The transcriptome is so much more altered than in compensated ASH. Something switches the transcriptome of the liver that drives the liver to fail. I always like the tissue to talk to me. I hate do research in which we predetermine which pathway or molecule we like the most. No, do omics, agnostic or prioristic, whatever, exploratory, let the tissue talk to you, tell you what pathways, which molecules are more deregulated. It's the way we do translational research. The most upregulated pathway in ALHEP versus early ASH, for example, is the EMT, epithelial mesenchymal transition. That is something that we can discuss. I'm not an expert in EMT, but I think, and I believe, and we have data, MADIL has also data in the recent JCI that a kind of EMT is occurring in the liver of ALHEP. I will show you data suggesting that. And this is one of our missions to reverse this phenotype to a normal epithelial hepatocyte. And what is the most down-regulated pathway? The bile acid metabolism. I just show you very quickly, this is the EMT genes. There's a lot of collagens, a lot of bimentin, a lot of markers of myofibroblast. They are here, okay. They are here upregulated more in ALHEP than in never decompensated ALD, etc. But we have a lot of biomarkers of EMT. And in the next slide, we can see the bile acid metabolism. There is a lot of differences between ALHEP and control of never decompensation. See how similar they are, a normal liver to ASH that never decompensated, and what a massive deregulation happens in ALHEP. We see here a lot of data showing a lot of downregulation or upregulation depending on the apical or basal membrane hepatocytes. All this data, of course, is available to any of you guys that want to do any study, for example, why these patients get jaundice. What a fantastic field for young people to focus in future research. Okay. So what about the ductal reaction, the famous ductal reaction? I remember when Pau Sancho returned from Belgium to our lab, and he's done a fantastic career in EDBAPS. We're discussing, hey, Pau, let's look at the amount of ductal reaction, stem cells, etc. in the liver. I bet there are none of these cells in ALHEP. We found the opposite. The more you have, the more you die. The more keratin 7 you have of all the markers of stem cells or progenitor cells, etc., more you die. So, as I say before, when you have such a good biomarker of death, either is a good surrogate of a bad outcome of hepatocytes, but also don't rule out that is a player. In other words, a good biomarker challenge that as a player as well. Could be a target for therapy. We think the reversing or stopping deep proliferation of these cells could be beneficial for our head. These cells are crazy. So look at this study that we collaborated with Philippe Mathurin group from the Excellence. Philippe group in Lille published this New England in which they offer early liver transplant in patients with ALHEP. Look at this slide here. It looks like the pseudotumor. My last slide of my conclusions is a little provocative about our new hypothesis, a paradigm or working hypothesis to treat ALHEP. You think these cells can allow blood flow to go nicely there? These are a massive dedifferentiation of hepatocytes towards a fibroblastic EMT or cholangiocytic phenotype. We need to stop that. If you don't stop that proliferation, no way the patient will survive. Okay. This is a study that I'm showing you unpublished data was published by from our group, and Meritxell is the first author. And now we are collaborating with the group in Barcelona, Garcia Pagan, and more people with Biobrom. We found something fascinating to me. Is when we correlate collagen with portal hypertension, it correlates very well in the alcoholic hepatitis. What about the pre-cirrhotic, the ones that don't even have advanced fibrosis? We have a very nice correlation. In humans, it's not easy to find such a correlation between the amount of ductal reaction cells and the degree of portal hypertension. And you look at the findings, 20. Can you imagine a 20 or 23 portal pressure in some or gradient in somewhere with advanced fibrosis with hepatitis B or C? Is unthinkable, no? So that's why the fibrosis classification has to be completely challenged in the field of alcoholic hepatitis and probably NAFLD. When we found that, what we did with Meritxell is let's do with the help of Pau, say let's look at the macrodissection about did with these areas. Maybe they have more vasoactive substances. Maybe they are releasing angiotensin, endothelin, vasopressin. We didn't find anything like that. What we found, they produce a lot of profibrogenic substances, and when we check for in the green is collagen around ductal cells. When you see yellow is that they co-express keratin 7 and collagen 1. But you see a lot of proliferating myofibroblasts, probably stellar cells around ductal cells. So ductal cells probably obstructed the flood by themselves. This is difficult to demonstrate, but they created a lot of fibrosis around, potentially producing and favoring portal hypertension. Okay, so this is something that a case report that typically in a scientific talk, you never put a case report. But this is a little link to my last slide that will be a little provocative, that already tweeted it, so is there. That is to see ALHEP in young people as a pseudotumor. Okay, so this is a case of a patient that they called me to the ER. So Ramon, we have a young lady with a massive tumor of the liver, is very yellow, And the first, of course, I'm biased, as you can imagine, but the first neuron that pop up in my brain is, this is an ALHEP. You want to bet? Went to the room, even from outside the room, I could see all the signs of alcoholism, stigmata in the face, etc. And I could easily say that was a patient with an alcohol use disorder. When look at this imaging, it seems like a massive, or a big liver, not crazy spleen, not a lot of, some collaterals for sure, but it was not crazy splenomegaly. But after stopping drinking, that reversed very much. When we did the biopsy, we saw an ALHEP with an early cirrhosis, with the bands were not very avascular, and ALHEP with all, you see here, the Mallory Weiss, Mallory Denk. And again, look at this proliferation of these keratin 7 in a patient that was reversible. Can you imagine this proliferation in these patients who do not improve with prednisolone of just abstinence? So that is a sign that ALHEP in young people is a pseudotumoral disease. It's amazing. It looks like a tumor. Well, you don't see any, in all these years, I have never seen a piecemeal necrosis in any ALHEP that was not taking cocaine, by the way. Never. So it's a very immunotolerant liver with this growth. Okay. What about the systemic inflammation that I mentioned before? We did a study with Javier Michelena and Joan Cabelli also helped us, and Pere Ginés and all the group in Barcelona. We have a very good, well-characterized cohort in Barcelona that I am missing and I will have again, because when you have a center that is so well organized with all the, I had to say that all these studies with all the group of Hemodynamica with Garcia Pagan would have never been done. So thank you so much for your collaboration. So when we did this study, we found in ALHEP that patients have leukocytosis, a fever, and not always are infected. I know the traditional ways to detect infections are not so sensitive. I know it. And you can criticize that some of them could have cold infections. Well, what we found, that patients with SIRS, not everybody is infected. Not in everybody we find an infection. In patients with infection-associated SIRS, they die much more than patients without SIRS in green. But there was a third subgroup, patients with SIRS without infection, sterile SIRS, similar what happens in pancreatitis. You can have a SIRS, you can have leukocytosis, and even low-degree fever without clear infection. And that killed you in 3 months, eventually the same. So no matter the SIRS come from infections or no infection, it will cause you multi-organ failure or death. So another line of research in ALHEP is what causes SIRS without infections, infection-associated SIRS, which cytokines, can we block them in a safe way? I have to say that not all cytokines are so important for infections as TNF. We can safer block interleukin-6, 1-beta, and don't cause as many infections as with ALHEP. So we have to find the sweetest spot in which we can inhibit cytokines but not promote bacterial infection, which is always tricky. Okay, so with all this data, we kind of revised a little the paradigm of ALHEP in 2020 to say severe ALHEP is characterized by inefficient hepatic regeneration and a systemic inflammatory response. Therefore, we need to improve all things to improve the outcome of the patient. Okay, the second part of my talk will be a little more molecular and the integrated omics. How can we identify such molecules or pathways? So to be honest, I don't see any other way than to have different layers for different phenotypes with different compartments, as I said you in the introductory slide, sorry. This is a study that was done here in Pittsburgh with collaboration with a lot of people, with EDBAPS and the STOPA trial people and BSA from the Mayo, a lot of people. And Jose Maria Argemi, that is now Maplona, was the first author. We did many different phenotypes, different omics at different layers. We did from RNA-seq to liver-methylome to ChIP-seq. We did with the STOPA with Atkinson and my Thursday US. We did with Helena Mann. She helped us to study the methylome. The Mayo Clinic people, Scott and BJ, help us to analyze the ChIP-seq. It was a highly collaborative study. Otherwise, it's impossible to do integrative omics because you need a lot of experts. So when we did this study, we did the following. I know this is a little busy slide, but let's focus in the last panel. This is the prediction activation, the predicted activation of transcription factor in normal people versus people that drink, but the liver is hanging out there well, no liver failure. The most upregulated, the most active transcriptive factors, it was super interesting to me, is PPR gamma, our endogenous statin. We have a protected guy, well known because a lot of drugs try to increase PPR gamma, were probably protecting us from liver disease. What happens when you do Al-Hab versus a drinker that was doing well? Where is PPR gamma? Now is the second more down regulated. To me, this is super interesting because it tells us that maybe PPR gamma agonists are good for clinical trials in patients with early alcoholic liver disease to preserve the liver function. It's not fascinating when you do omics. If you're an MD, you will enjoy a lot reading the results. It's the most fun part to do research. But as I told you before, we focus in the data that the tissue tell us. We don't tell the tissue what we want to find. We hear and we read the most downregulated transcriptive factor whose transcripts or genes, dependent genes, are more downregulated in Al-Hab is HNF4 alpha, probably the main governor or epithelial cell function like albumin production, clotting factor production, production of key genes that encode enzymes involved in glucose metabolism, for example. They were really down. That's why we focused on that. The first thing we did, I'm not showing the data, I promise you not to show too much data, is GWAS and say maybe polymorphisms in these HNF4 alpha or 1 alpha predisposed to Al-Hab. And with the help of Steve and Mark from the UK, we found negative results, nothing. In fact, I envision that based on these and other data, that Al-Hab is not such a genetic disorder. It's mostly an epigenetic problem, okay? So we focus why HNF4 alpha genes are so downregulated. Is that epigenetic if it's not the GWAS? So we did ChIP-seq, I'm not showing the data. We show that clearly the chromatin was closed in the areas where HNF4 alpha should bind the DNA. What about the methylomics? Look at these, and I promise this is unprioristic. With the help of Helena Mann, we did analyze these. We did methylomics in the liver and Al-Hab, and we say, which transcriptive factor has the most hypermethylated gene? You know, hypermethylation will prevent a gene to be transcribed. So we found that the number 1 and number 2, this is number 1 and 3, and this is 1 and 2. The 2 transcriptive factors which genes are more hypermethylated are HNF4 alpha, 4 and 1. Look at the number 3, by the way, PPR gamma. So that's why part of the research we're trying to do right now is which epigenetic modulators change in Al-Hab. Maybe epigenetic modulators, there's a lot of drugs, can play a role in the therapeutics in these patients, okay? So let's show you a couple more data, okay? After we complete this study, we're very excited about glucose metabolism because glucose metabolism is tightly regulated in the liver. A healthy liver is the most generous livers for the glucose. Doesn't use the glucose, store the glucose. The exokinases are not very avid to phosphorylate the glucose. It just keeps the glucose for the rest of the body. In Al-Hab, it becomes a greedy and misusing the glucose. I will show you some data on that. And that could be also a target for therapy to try to improve hepatocellular function. So this is another I don't have time to go into detail, that was done also with collaboration with Barcelona. This Veronica Mesa is the first author, Jose Maria, Merichel, and a lot of the people here, Anna from our lab, a lot of people play a role. But we have different phenotypes again, normal livers, alcoholic hepatitis, alcoholic cirrhosis. I had to say that the normal liver is the most difficult phenotype to find good livers, by the way. And we did liver metabolomics, liver RNA sequencing, also ChIP-seq, etc. I will show you these 2 layers. As I say, different phenotypes, different compartments, different layers. Remember that. We did metabolomics in the liver, knowing the blood, and liver and RNA-seq and integrated the data. Let me show you the data. This is the results of the whole big cluster in the liver metabolomics. There's always some outliers in the human studies, but they cluster very nicely normal, alcoholic cirrhosis, and alcoholic hepatitis. And you see that they classify very separately, except for one. alcoholic hepatitis and alcoholic cirrhosis. Okay, so when we look at all the metabolites that were differentially regulated in the three phenotypes, we decided to focus on carbohydrate and glucids because we found them really differentially regulated, and we needed to focus on some. Remember, for collaborations, we have data of many other lipids, amino acids, etc. And I always feel the omics data is a pity to put it in a drawer and not use it for multiple studies beyond what a PI can do. So please contact me if anyone wants to do any collaborative study with these omics. This is the RNA-seq data. The RNA-seq data from control livers, alcoholic cirrhosis, and alcoholic hepatitis. And I'm showing here genes that code enzymes, key, and glucose metabolism. Look at these. Glucogenesis, it was really down in our head. Glucose lysis also was down, and you don't store glucose, but the pentose phosphate pathway, that is an alternative way to phosphorylate, use a pathway typical of diabetes. It causes oxidative stress, it causes sorbitol, that causes also osmotic stress. It's a bad way to metabolize, glucose. Okay. So here we try to integrate, okay? And it's always difficult to find bioinformaticians that integrate. This is one of the people that I'm chasing the most, integrators, both clinical and omics, both different layers of omics. When we integrated that, in blue, you see the integration that is down. When you see glycogen storage, and it was really down. And the same way the Krebs and the ATP, etc., production of the normal glucose metabolism was really down. Was this really up? Was the abnormal glucose metabolism, in which we found a new exokinase that is very avid to metabolize and to phosphorylate glucose. It's not generous the liver anymore. It's using glucose for itself. It produces fructose, which is known to be deleterious to the liver, but also it produces ROS, sorbitol, etc. So we know that maybe targeting some of these genes could restore the normal hepatocellular function and normal homeostasis in alcoholic hepatitis. And I will show you that, just to finish my presentation, to a couple of slides of the latest we're doing. We're writing now this with a study. Everything that I have shown you so far is bulk liver, the whole liver. And for many years, we have been very speculative interpreting bulk liver results. I don't know which cell types are producing what. Of course, we can do immunocytochemistry at the protein level, but we can do immunocytochemistry of 20,000 proteins. That's why the single cell has become like a great field to do cell-specific expression of genes. So this is a study that Steve Atkinson in my lab is leading, and we are preparing now the manuscript. We are very advanced. Steve is I think is a brilliant guy. I have an idea, Ramon, and let's first do an in silico of a published paper in Nature Communication for the UK. People are a friend of mine, they gave me the data, the raw data. They have progenitor, cholangiocyte, and hepatocytes. We need to identify a signature of cholangiocyte and a signature of mature hepatocytes. And they check our ALHEPs, all our cases where the cells rely on. Are they more pure cholangiocyte, pure hepatocytes? Where are they? Using these signatures to define what is a pure cholangiocyte versus hepatocyte. This is a summary of our RNA-seq data. We did RNA-seq, from single cell, from healthy livers, NAFLD, PSC, alcoholic cirrhosis, and alcoholic hepatitis. I have to say that in Pittsburgh, that we have the most active live donor program in the country, and one of the ones in the world, has helped me a lot in this research, because having normal livers from donors, having all these explants, and also there are scheduled therapy surgeries in the morning, is not in the middle of the night. So thank you to Dr. Umar Molinari and Hughes, because without the help, this could not have been done. So this is the colors of different diseases, and this is the hepatocyte score that I showed you before, and cholangiocyte score. Nothing changes so much until ALHEP. Look at the ALHEP. The blue is ALHEP, severe ALHEP. So they have less cells with the pure hepatocyte score and more cells with the cholangiocyte score. You see that? Hepatocyte score is down, cholangiocyte score is down. When you put all together here, in yellow, it will be epithelial cells. In orange, hepatocytes epithelial. In adult progenitors, in purple, and in dark purple blue is fetal progenitors. Okay? Look at LHEB. LHEB have very little amount of cells that retain epithelial pure hepatocytes. The good hepatocytes are gone. Most of the hepatocytes are now a mixed population of cells that express a lot of biliary. It's the ductal reaction that I told you before. So most of the hepatocytes are transformed into a cholangiocyte-like phenotype that we don't need. The normal liver doesn't need so many tubing. Cholangiocyte, the tubes to the blood flow. You don't need so many tubes. You need bricks. You need the good bricks. You need the parenchymal cell. You need hepatocytes. So we are analyzing, of course, different genes, different markers, unique of different phenotypes among the hepatocytes to identify potential target for therapy. And as a conclusion of the whole talk, instead of having the typical boring bullet points that there's always a summary, one day I was a little high and one say, okay, we need to change our mindset in install HEP, especially in young people. And based on these studies, let me be a little provocative. And I view HEP as a pseudo-cholangiocarcinoma. That's why we're very excited to help with cholangiocarcinoma experts now with Silvia Fo and Pau and other people. And Jose Maria would like to work with him as well, because I want to know something that applies to a cholangiocarcinoma in a lesser extent could apply to LHEP. Can we revert that to a normal hepatocyte? When we need to reduce proliferation of biliary cells, and we need to return hepatocytes for a cholangiocyte phenotype to a epithelial albumin-producing, clotting factor-producing phenotype. So I just want to thank, this is a super collaborative study from many, many places. This is from my lab, but the collaborators for Idibab Pau has been a super loyal and so helpful, but I don't have time. But also I want to say that Juanjo Lozano from CIBRED has been amazing, has been so helpful, and the CIBRED people that they are listening to me, please take care of Juanjo because it's been amazing. And a lot, many collaborators. Thank you for your attention. This is, number one, very difficult question, very important. Number one, the same way people don't eat the same 1 week, they don't drink the same all the time. I don't know, we have to say they drink the last month, lifelong drinking, area under the curve of taking all their life, or pattern. One of the problems, there are two problems in quantifying. Number one, patients lie and they report all the time. The stigma, the shame. We need biomarkers. We have a very good review with Joaquin about biomarkers that you can read in Clinical Liver Disease, no, Joaquin, I think?
Yeah.
And I will highlight the PEth. The PEth has changed our life here. And I would like to bring into Spain because I don't think many centers are doing it. It captures 1 month of drinking. You can cheat with that. It's not urine-based, etc. The second thing we need, by the way, I'm trying to start an initiative with that, is a universal way to measure alcohol. Each country, each continent, each place is units, grams, liters. The units doesn't mean the same in Australia than UK. We need a universal, in a global world, we need an universal unit to measure alcohol. Not a difficult question. None of the questions are easy, but let's say you have a fat person that have metabolism for many years, that they compensated cirrhosis, and you suspect he's drinking. You do a biopsy, and then the biopsies can be similar. I have to say, though, that alcoholics have metamitochondria, probably more severe. Mallory-Denk, more severe. But the only pathognomony probably is megamitochondria, and the distribution of fat maybe is smaller than the NAFLD. But they can be superimposed. They can be the same. That's why we need more molecular markers, okay, to distinguish that. Analytically, ALD, when they are drinking, the GGT goes to 200, 300, 400. When I see an NAFLD with a GGT of 400, my red flag, okay, pops up from my brain. So Eddie, I have to say, I don't know, in Twitter, that is, by the way, I have never been so updated, because Twitter, there was a study last week about the epigenetic scar after curing hepatitis C. You cure hepatitis C, and hepatitis C causes you so many epigenetic changes in your immune system that despite curing the hepatitis C, you will always retain some epigenetic changes for life. I felt it fascinating. Nobody knows in ALHEP. I wonder whether some of the changes are reversible, and some of them will maintain in time. That is a very good question. I think Edilmar is doing now a study with sequential biopsies, etc., and we can personally address this interesting question. We're working in cell-free DNA methylation patterns now in ALHEP, and especially because knowing that some of the genes are very hypermethylated, and like HNF4 alpha dependent genes, maybe we can check not only in ALHEPs, but also in clinical trials or in compensated alcoholics, whether some patterns, in other words, if your genes that produce albumin is started to be hypermethylated and you detect in the blood, despite having a normal albumin, is a red flag. Your machinery producing albumin is started to be damaged by alcohol. Be careful, you don't stop drinking. The next step will be hypoalbuminemia and ascites, for example. I'm just making an example. Yeah, that is, remember with Joaquin was in my lab, remember in Chapel Hill, we wanted to do, and we study living, because of metagenomics, not human, by pathogens in the liver. We were very excited because we detected hepatitis C in the hepatitis C patients, etc. But we basically found a lot of gram positives, but we found more in the same in the controls. There was basically contamination from the extraction. It's not sterile sometimes when they put the liver outside. So I think, yes, I think there is more data showing that PAMs and not only PAMs, part of bacteria, but viable bacteria can be detected in damaged tissue. And my goodness, the liver is so exposed to the portal vein that I bet this should be one of the next studies to be done in very carefully handled tissues to avoid contamination. We found a lot of gram positives, that I think, because once the liver is out for research, you're not sterile anymore. Okay. Very good. Thank you, Xavi, for this interesting question. In fact, this is you can do the deconvolution of the data. There is some software that, based on previous pattern described for different cells, they can infer more or less the cell types that are responsible for the bulk RNA. In fact, if I'm not wrong, Laura Nagy did that in silico study with a bulk RNA using this technique. I think, I don't know where she published that, but they published, I think, that deconvolution model to predict. But I think, to be honest, if we have now single cell, I believe much more the single cell data than know any models involved, to me. So I think we should move to a more single cell data. I'm not an expert in mitochondria. My knowledge is very limited. I know Pablo is from Madrid, is studying now mitochondria. There are other groups. The study from Maturin Philippe did microscopic electron microscopy, and the mitochondria was really altered. Now Steve Atkinson is trying to find mitochondrial DNA mutations in the liver. Yesterday we have a meeting, we have some problems to detect circular DNA from mitochondria, but I think mitochondria in ALHEP should be another field of research in people interested in this organelle. I think I'm sure it plays a key role.
Thanks.
thank you, Giovanni, for asking me the question that I never like to be asked. You know why? Because I never like this question because I'm not an expert in cell origin. The experts in cell origins are very opinionated. When you say that they are even two theories, if they defend one theory, they will jump your jugular in the middle of the. So I think, if I'm not wrong, I will tell you my opinion, but I'm not an expert in cell origin, stem cells, etc. Based in immunocytochemistry study and cell single cell, I think in part oval cells coming from biliary ducts can raise to beta ductal erection cells, but I'm convinced, seeing all the heterogeneous and progressive de-differentiation of hepatocytes, that the adult hepatocytes also give rise to all these ductular cells. I'm convinced because I've seen a lot of immunocytochemistry of intermediate states of the differentiation. So, and I don't know how stable they are. This is beyond my expertise. I don't have a clear answer for this. These are people that develop much less ACC, okay? And it's true that they are younger, but as I follow them more years, they kind of protective. Very few patients that have developed ALHEP ever develop an ACC. And I have the theory that maybe is the immunotolerance, that it's almost like or the immune system. But this is just a speculation. We don't have, I have seen one patient in my whole life with cholangiocarcinoma and alcoholic hepatitis 1. None of the other patients, thousands of patients I have seen ever develop a cholangiocarcinoma. So in somehow, ALHEP produces half of the way to a cancer, but never the full cancer. Many of the pathways are oncogenes, many of the mutations are found in cancer, but ALHEP is a castrated cancer production. It's in the middle, and they are protective. I don't know why. Maybe it's epigenetic modulators that prevent very apt, and they prevent other mutations that drive cancer. I don't know why. ALHEP people never develop HCC or liver cancer. Never, barely develop. Yes, and I think bilateral irrigation, DDC, I'm not the main expert. You should ask probably Pau Sancho. He's much more expert than me in that. But yes, there's good models of ductal reaction. Yeah.
Okay.
But it's difficult to do that in a model of ALHEP. That is the problem. Yeah. When I say pseudo, is that, my goodness, you see a massive proliferation of bile-like cells. Let's say ductal is a massive proliferation that I remember when we were writing this with Philippe called it infiltration. I say, Philippe, this is not a tumor. We cannot call infiltration. Let's say proliferation. But we were discussing, Ramon, it looks like a cholangiocarcinoma. So if you don't they tell you it's not an ALHEP, you may think it's a tumor, because it's such a massive. And I don't know so much about how reversible it is because we don't do pair biopsies in these patients. I think the UK, in India, now Eddie in Sao Paulo, there are some people starting to do serial biopsies. We can answer this question. What is reversible, what is not? That would be very cool to do serial biopsies to see what is the reversibility, and now if we can predict recompensation of the patient, need for transplant, etc. Yeah, so the problem of the models that I mentioned one of my slides is a difficult one in this field. The max you can cause in mice is early ASH, that's it. To cause ALHEP, we have given CCL4, DDC, Ivan Rusin, I know Paul has done other efforts, other people, transgenic mice. It's pretty much impossible to cause that in mice. That's why we're shifting a little bit to human-based tools, because I need to, at my age, and you can see my gray hair, I'm not 20 or 30, I need to believe what I do, not only to convince the reviewers, I have to be convinced myself what I do is meaningful. And I don't think I'm doing meaningful preclinical studies in ALHEP mice. I have to be honest. when you don't have any portal hypertension, any massive fibrosis, no bilirubin no liver failure, how can you translate that to humans? Not the differentiation of hepatocytes. So I'm not convinced that these models are good based on the data. Of course, this is a great question. I think everything is linked, and there's a lot of perisinusoidal also fibrosis in ALHEP. There's a lot of capillarization in ALHEP. There is some data from Bersnabel, from Bao Sancho, from BJ Sa, that no more angiogenesis than me endothelial dysfunction showing endothelial dysfunction in ALHEP. But I'm sure this is worth it to do studies. The good thing of ALHEP, everything you study is so altered that you can produce good papers, because the null hypothesis will never be a fact in ALHEP. Everything is so altered that is worth it to do studies and correlate with the outcomes. So I invite you to proceed with this research and this question, very interesting, but unexplored, relatively unexplored. There is few data in exposome. Exposome includes pattern of drinking, the way you drink, if it's illegal alcohol or homemade alcohol. And the exposome includes if you drink with it's like gin tonic with a lot of fructose, or you are obese. There is not much data, but I think a lot of research should be done. Why some people develop ALHEP versus others do not develop ALHEP. Part of the explanation could be the exposome, but it's unexplored, and I invite you and many others we need to collect better the exposome to do correlation studies. The exposome so far has been suboptimally collected. That's why we don't have enough data to do robust studies. Thank you. Very important questions. Of course, the cells are dedifferentiated, but some of them is necroapoptosis, pyroapoptosis. There's different mechanisms of cell death. The other experts know more than me in that. I'm not an expert in cell death mechanisms, but there are many studies showing that alcoholic hepatitis is also associated with cell death, apoptosis, necroapoptosis, pyroapoptosis, induced by iron as well. So this plays a role for sure. Two comments on that. Number one, in silico, I promote people to do in silico. We put too many papers, too many raw data in public, and it looks like people don't want to use it because it's no, if the in silico data is well done, is a different approach from the original data, can yield fantastic papers. I have seen Nature papers just based on in silico studies. Okay. Number second, you're on point. Predicted something doesn't mean that the actual genes were differently transcribed. And we have to be very careful in differentiated predictive models to models in which you generate or you study all target genes. I have to say that many of the target genes that we have studied in ISFOR alpha, both peripherically on the liver, confirm our results. And clinically, a lot of genes, albumin, for example, factor 7 and 5 of coagulation, they are really done. But I'm on point. One thing is to do predictive models. Another thing is to do the real deal. Okay.
Thanks, very, very thanks, Ramon, for this fantastic seminar. See you next seminar, next liver seminars.
Thank you, everybody, to attending.
Thank you, everybody. Thank you, Ramon.
See you soon. Take care, everybody.
Bye. Bye.